La siguiente, que destacará la importancia de estar preparados para futuros ensayos clínicos. Quien está a cargo de tan vital tema es el doctor Laurent Servais, profesor de pediatría y neuropediatría. Tiene una destacada experiencia con enfermedades neuromusculares pediátricas, la abreviación es MDUK de Oxford, es el Centro Neuromuscular y es profesor invitado en Neurología Infantil en la Universidad de LIEJA. Espero haberlo pronunciado bien. Así que muchas gracias, doctor Laurent Servais, por su tiempo y terminamos con esta última conferencia.
Hola a todas y todos, mi nombre es Laurent Servais, yo soy profesora de pediatría y neurología a la Universidad de Oxford, en Inglaterra y también en la Bélgica. Me gustaría hablar castellano durante toda la conferencia y yo trataré durante las preguntas, pero por la lectura me voy a hablar inglés, que es un poquito más fácil para mí. So, today, I'm quite excited, to be honest, because I should not tell you, but we included our first patient in a therapeutic trials for Angelman. And so maybe I should not say that, but I'm so happy to share it with you for a very simple reason, because it goes so well with the title of this talk. This title of this talk is Being Ready for Clinical Trials. And to a certain extent, we were ready. Maybe not as much as we wanted, because you're never completely ready for clinical trials. We've been running clinical trials in neuromuscular diseases, in Duchenne, in spinal muscular atrophy, in X-linked myotubular myopathy. I can tell you that Angelman is a different challenge, but a very exciting one. Actually, when we speak about clinical trials, most of the time we are very excited. We feel that we have in our hands the drug of tomorrow. But we must be clear with the patients. We must be clear with the parents. We must be clear with the community. Trial is a trial, nothing more. Trial is something we do to check a very simple question. Does it work or not? Is it safe or not? What is the best dose to give, right? We don't conduct trials to treat little Miguel or little Juanita. We conduct trials because we want an answer to a question. And unfortunately, for instance, in Duchenne muscular dystrophy, a disease I've been very active in, I have a long track record of failures. I've seen so many trials that failed. In spinal muscular atrophy, we were a little bit more, lucky. We had more success. And sometimes I try to figure out actually why we were so successful over the last years in spinal muscular atrophy and not in Duchenne muscular dystrophy. Of course, there is a matter of the drug, right? I mean, whatever is the trial, whatever the trial readiness, having a drug that works, that helps. But nevertheless, if we want to be successful in our clinical trials, and I would say even in our clinical development, because a trial is just a piece of the puzzle of a large story that is the clinical trial development, we must check the boxes, we must be ready. So one of the first things when you start, preparing a fight against box, for instance, or in any kind of fighting is to know your enemy, right? You wish to know against who you are fighting. What is our strength? What is our weakness? And so it's very difficult to start trials if we don't know the disease that we're going to challenge into these trials. Well, we believe that we know the diseases because we are very clever doctors, right? But when it comes to say, at what age do the patients start having the first seizures? At what age do the patient lose ambulation when they do? At what age do the patients say the first word? What is the number of words that they say if they have a mutation, if they have a deletion, if they have parental disease? All these questions, when we need a very precise answer, we don't have it. which means that when it comes to try to figure out what to measure when we conduct a trial, this is another story. And when we have something that is successful, and we all believe as a community that it's successful, when we go to the payers and they say, well, you know, are you really changing something to the life of the patient by changing this or that on this or that scale? We need to have data. And this data, we can acquire them during what we call natural history study. So that's the first thing I started in the UK when I arrived in the UK 2 years ago, just before Brexit and COVID-19. Can you imagine how crazy I am to come in the UK just before Brexit and COVID-19? So we started a natural history study just to try to understand what is the journey of these patients, how many patients we have to conduct clinical trials, with which mutations. And actually, if 2 years ago I had to start clinical trials for Angelman in the UK, it would have been impossible because we were not ready. So as I told you, I had this experience with different disease, and one of the diseases, spinal muscular atrophy, for instance. The nusinersen, that was the first drug that was approved by the FDA, was approved after a double-blind randomized placebo-controlled study. But this double-blind randomized placebo-controlled study was organized on the basis of a natural history. And we could not have defined the endpoint of this pivotal trial if we had not conducted before a natural history study. So the natural history study in spinal muscular atrophy, which is the paradigm of success over the last year for neuromuscular disease, was super important to identify potential endpoints, to identify potential sites, to establish early relationship, to engage with patients community, and to engage with potential biotech and leverage resources. So how will it work? Our clinical trials, our natural history study, and when I say ours, it's just because we wrote the protocol, but we have shared this protocol already with several other investigators. We will enroll about 40 patients. They need to be younger than 99 years old. If they have 100 years old, if they are 100 years old, we will probably not include them. Just joking, but not that much. I know there are no patients around who are 99 years old, and I know that if by chance we identify patients who is 50 years old, probably a clinical trial with a drug will not be initiated in such patients. But nevertheless, we have a lot to learn from all the patients about how the disease has been progressing over all these years, what has been the burden, what has been the cost for the society. And you will never know by including a patient who is 6 months old what could happen at the age of 10 or 15, 20 years, but you can know it with all the patients. So even all the patients may bring retrospective information that is extremely helpful to understand the progression of the disease. So of course, we'll start with the baseline and questionnaires to the carer, a 2-week sleep and seizures diary, a medical assessment, blood sample for biobank sample and biomarker, 24-hour EEG, several questionnaires. And we will use a device that is now used in several clinical trials, that is Actimio, because we now have the possibility to record many more informations using wearable sensors that do not disturb the patients, but that collects very large amount of data, and that collect the real life data of the patients. And then we'll follow up on a regular basis these patients. So that's the wearable I was talking about. It's like 2 watches that the patients wear on the ankle, and it can record the stride speed, it can record the perimeter of walk, so what is the longest distance that the patient is able to walk, the number of strides per hour, what is the activity of the patient, the number of falls, all of this can be recorded. And of course, if we record during 2 weeks, during 1 month, we have a very good understanding of the patient conditions. We have a biomarker study for EEG, and we have EEG. We use the scales that are used now in the most cutting-edge trial, like for instance the ORCA, which is a scale that has been developed especially for Angelman syndrome for communication. We use for maladaptive and aberrant behavior, ABC, and Vineland score, and several motor scales and quality of life burden. This is extremely important from a payer perspective, because one more time, it's not enough to have a drug that works. It's even not enough to have a drug that is approved. We need a drug that is approved and that is reimbursed for patients. And for that, we need to be ready to explain to the payers what is the benefit and what was the situation before we had the drug to deliver to the patients. So as a matter of conclusion, I would say a natural history study, it helped to prepare clinical trial readiness. Because if you know that you have 40 patients, 25 with the deletions, 6 or 7 with the point mutations, and you know their age, it's super easy to include a patient in a clinical trial because you know where they are. You know they are committed to come in the trials. You have had the opportunity to meet them, to discuss with them trials. You know who you're talking about, which means that when you discuss with the company and you say, I can include exactly 6 patients between the age of 6 and 15 years, or with deletions and stable seizures. You can just know it very easily if you have a clinical trial, natural history study that is ongoing. But more importantly, it helps also to appreciate the burden of Angelman in your country, in your region, and to provide payers with data, because sooner or later the question will be, is the drug reimbursed? It accelerates patients' inclusion, as I told you, and it helps to validate outcome measure. The validation of outcome measure is a continuous process. We should not think that because an outcome measure has been validated, it's good forever. This is not true. We need several studies. We need larger database, and we need to, at one point, to be able to understand what means a change over a short period of time or a longer time period. Because today, we have 12 drugs that are reaching clinical trial site. And the question today is, any of these different drugs, how they compare to placebo or to the absence of treatment? I wish that in 5 years, in 10 years, the question will be, how do they drug work together? Can we push the envelope? Can we hope that by associating treatments, we get a better effect? And this kind of question will be much more difficult because it's not the difference between white and black, it's the difference between different shades of gray. So it means that we will need at that time even more powerful outcome measure to understand how a change in a short-term period translates into a change on a lifetime period. And we'll have better outcome measure to discriminate small changes in short period of time. So this is the very first step. There are many others that we need to get in place to be ready for clinical trials. Clinical trial readiness is a continuous process that involves not only you as a doctors, but all your team and all your environment, your hospital, your administrative. This is a fascinating journey, sometimes a little bit scary, but with so much rewardness, because I can tell you that as today, when you include a patient in a therapeutic trial, you feel good. It was a pleasure to be with you today, and I look forward to any of your questions.